Hello and welcome back to chapter 2. So let's welcome you to our tutorial on UniProt. If you were to Google UniProt using google.com, you'll be able to see the results here, and all you have to do is just click through and you will be directed to directly to the UniProt database. As you can see, this is a very user-friendly graphical user interface, and what we're going to do today is just a tutorial, a very simple one, on how to search certain things and how to look around at all the information related to the keywords. And just a brief idea of what UniProt is all about. There are a lot of sections and buttons here. So just so you know, anything that you search by UniProt, you could just look it up and look at all the sequences in the database. You can retrieve ideas or any list of peptides from this database. And if you're always doing protein identifications, then you could use UniProt to submit peptides that you want. So basically, when people use genome assembly annotations and use sequences from UniProt, because in UniProt there's a lot of functional and annotated genes. What you see from the website is a number of records that are currently available, that are stored. And this is periodically updated, and later we will see that there are a lot of number of records that are searched. Just to know, if you scroll down, you could also see that you could submit your data. So if you're thinking of submitting your data and then publishing it on UniProt, you could actually just come to this website and click the Submit Data button. The rest of it is pretty much general. The only thing that we will actually look around is how to do a tag search using any keywords, and come to use Blast through UniProt. So if we type in a keyword, collagen, and press enter, then this will bring us to our search results screen, and you can see this. All the results are based on the keywords that you have entered. There are two sections here. The first section to the left would be all the links which are associated to the keyword search, and under this link, which was filtered by under two types of informations which have been stored in UniProt. The first one are those records which have been reviewed by scientists, and those records which have not yet been reviewed by the scientists. So in this case, if you see any symbols with the yellow color, then that means that record has been reviewed, and if it's blue, then it has not been reviewed. Yeah, so under this section you'll see similar genes under different species, but we're just focusing on humans in this case. So for now we're just looking at humans, so now we're just clicking human here, and then we could only see there are 269 collagen records, 500 from human. Yes, and here you can see collagen associated with other types of keywords, such as disease, domain, gene names, gene antigens, and other kind of records. So if you want to like what type of genes for collagen, but maybe some of them are just looking for the protein family, they could look through protein family down here. In this case, I just want to click disease to look up the records. Right, now once we click disease, we could actually disease the functions of this gene would be is to accelerate the folding of proteins. It catalyzes the cis-trans isomerization of proline unique peptide bonds in oligopeptides. So what else can you actually see? If you scroll down, you could see there's some help there. That's the links to the information. Now, let's focus on the second part of these retrieved results, which is the results itself. Remember, as a good database, you need separate sections of the table. So each of the table contains specific information related to one. So in this case, you see a real table here. This, which includes entry name, protein name, length, pathway, the function associated to this, as well as protein interactions in our entry. We could actually change this whole database, the way it views the results, or what columns to show. How do we do that? It's very simple. You just click the button, and then you are able to add other columns that can be displayed, and you can drag them wherever you want. For example, if I want to use move around interaction with to the end of the table, I could just move it to the end. I could also pick or click, tick other options to show with the checkboxes, so we could click them and pick them and save those results to show in the table. For the time being, I don't want to see the link, so I just uncheck the checkbox and I remove it. And then I click the save button, and then we'll not be able to see the length of the gene anymore. Now, what else do we want to see? So let's play around with this. I want to see function. I want to see pathways. I want to see functions. I want. Let's look at some other ones. Let me see. I want to see add tissue specificity. So I'm just going to randomly click on different ones and have a play around and have a look around. So I'm just randomly clicking all these things and save it. And here we are. So now we have a number of columns here added to the table. There's a lot of added information down here. Right. Okay. Okay. Right. The good thing about Unibroad is that if we want to do sequence alignment on a blast, that's quite easy. You just have to click information that you want and then click the blast button. Once you click blast, then it will tell you whether you want to do a normal blast or a specific, or you could do a specific parameters that you want to use for this process. In this case, I just want to show how it goes. So you press go, and then it will do its thing. And then it will take some time depending on the length of the sequence as well as of the speed of the internet connection. And now, meanwhile, we could just see this is the example of the sequence belonging to peptides, polymer, cis, trans, Homo sapiens. And you could, if you need help, you could look at the BLAST help video or check out the tutorials. Or you can download the results of the BLAST. So that's it. So once BLAST results is out, it looks like this, and it tells you in a practical view, if it's red, it's 100% identical. If it's blue, it's towards the less similarities of the sequence. Now, the most important part would be when you get this kind of results, you can just arrange the way the results are presented by the e-value or the score. So, yeah. So just checking it out. There's all kinds of records here. Okay. Okay, I guess that's about it for now. I hope that helped in looking at UniProt. It's my first review of this, and we'll need to